Okay, so in this recording I'm going to show you briefly what crystal structures are and what they look like. So I have open in front of me a PDB file. So this is the end result of the crystallography process that we upload into the database, and it is just a text file. And the top part of this text file has a lot of information about the protein. So here's the name, angiotensin converting enzyme. You'll also have the PDB code, information about how the protein was expressed, information about where the paper was published, and then it will have details about the crystallography process and whether the structure is any good or not, which we'll look into later. So all this top part here, it says remark, it's telling you things about the protein, and that's the sequence just there. That's a list of the secondary structure elements. If we keep going, eventually we get down to the atom positions. Now in this case, if we look down this column here, we can actually see there are two types of records, something called atom and something called anisu. So just ignore these anisu ones for the moment. This is only something that some PDB files will have, but the important thing is to look at it where it says atom, and this it shows you basically the output of your crystallography experiment. So this is atom number 1, it's a nitrogen, it's part of a leucine amino acid, it's in chain A of the sort of, if there's like two proteins within the asymmetric unit, and leucine is residue number 1. And for this atom, what we have is 3 coordinates here, x, y and z. So this we could plot this atom in space using these coordinates. This is the occupancy, which don't worry about now, and the B factor, which we also didn't worry about now. But basically what I want you to see is that the output of our experiment is a list of atom positions, x, y and z, for all the atoms in our crystal. So if I just minimize this a minute, a much easier way to look at this information is to display your PDB file in a program such as PyMOL, which you'll get to use later in the semester. So this is, you can see here we have 2 molecules A and B in our asymmetric unit. Don't worry about that yet, but basically it means we've got 2 versions of what our molecule looks like. And if we zoom in, that's centering, and then dragging with the right mouse button down, what we can see is how these atom positions have been plotted. And also if you scroll the mouse wheel, you can sort of slab it so you don't have to look at all the atoms at once. And so here we can see represented, we have at each of the points where these lines meet represents an atom. So this is an alpha carbon atom in this residue here, which is a serine. You can see this is the serine side chain because it's got 1 carbon, and then the red indicates an oxygen. So this is a serine. This is the peptide bond here. Usually we have blue indicating nitrogen, and then this carbonyl oxygen here, also represented in red. And so we can possibly trace through this sort of long stick our backbone of our polypeptide chain, and off this we can see various side chains. Oh dear, I've clicked on the wrong thing, so that's not a very good one to look at. This one's a nice one because it's not surrounded by lots of other stuff. So this is an arginine side chain here, and again these are just plotted atom positions as points in this sort of ball and stick type diagram, but without actual balls and sticks. And if you're wondering what these little red stars are, these are representing oxygen atoms, which is all we can see of these ordered water molecules which are surrounding our proteins. So there's lots of water within the crystal, but a lot of it's sort of disordered in these large gaps, and so when we solve the structure, this water gets averaged out and we can't actually see it. But there are some water molecules that are actually hydrogen bonded to our crystal protein, In this case we can actually see them because this water is in the same position all the way through the crystal lattice, and it's probably forming a hydrogen bond here with this serine. So if we zoom out again, you can see actually how many water molecules we can actually see for this structure. I'll anti-slab it again so you can actually see it a bit better. And the number of water molecules we can see is actually dependent on the resolution of the structure. So this is at is it about 2 angstroms or 2.5 angstroms? No, 2 angstroms. And so we can see all of these little red dots surrounding our protein. At 3 angstroms you would see far fewer waters. So the great thing about PyMOL is you can actually then choose to display your protein in different ways, so we can explore different things about it that we might be interested in. I'm just going to see if I can find, because I know buried in here somewhere is a ligand. I can tell that because it's easy to find because it's actually got different atoms in. So this orange is actually a sulfur atom, and this grey point here is actually a zinc that's bound to the enzyme that's involved in catalysis. And so if we want to see this ligand a bit clearer, we can choose to display it differently. And if we right click on this atom, we can do show, and I know it's just an atom, so I'm going to say spheres. And rather than just drawing a point, what it does is it actually draws a sort of sphere to indicate where the zinc is. And if that looks quite big, that is because zinc is a very big atom compared to all these oxygens and carbons and nitrogens surrounding it. And this is a ligand that's actually bound to the zinc, and so we can also choose to show this in sort of more ball and stick format, so we can actually see the ligand better. And so what we've done is we've drawn this with sort of thicker lines, and this is helping us to see how the ligand is bound to the protein and the zinc. And the thing to notice about this is that we haven't changed any of our data. What we're doing is choosing how to display our data in a way that's going to tell us something useful about our protein or help us think about how it binds to the active site. So we can also do this change of appearance for maybe these residues that are also bound to the zinc. We might be particularly interested in them. There are other ways to do this with the command line as well, but for the time being this is a very suitable way to do it. Residue show sticks. And so now what we have is we have our 3 residues that coordinate our zinc ion shown in this thick stick, so we can see which ones those are. We could label them, and we can also see that we have this inhibitor, which you can see looks a bit like a protein. It's got and that's because it's based on a peptide, although it isn't actually a peptide, and that's bound here in the active site to this zinc. And then these are the rest of the residues in the active site. So that's one way we can choose to look at our molecule. Another way, which you should be quite familiar, is to look at the ribbon view. So if I do show ribbon, show cartoon. We call it ribbon, but it's actually cartoon in PyMOL. So what you can see here is the standard way that protein structures are often displayed, with their secondary structure shown as a cartoon. So again, what we've done here, we haven't changed our data in any way, but we've just chosen to display it in a slightly different way. And we can actually hide that's worked. Hide the lines that we had there before, and what we can see now is the ribbon showing the secondary structure, how the fold of the protein is, without having to worry about where all those little atom positions are for individual side chains. And this is usually quite a neat way to show the overall structure of a protein. Obviously we've still got all the waters displayed, but we wouldn't have to choose to display those, and usually we don't bother displaying waters unless they're particularly interesting for something we're looking at. But because we've actually chosen to show these as sticks, we can actually still zoom into the active site and we could actually just have a look at how the ligand lies in relation to these secondary structure elements around it here. We can also do things like, let me just check I've got the right one, residue, change the colour. It's always useful to keep it coloured by element, which means that the oxygens stay red and the nitrogens stay blue, but you can then change the colour of your carbon atoms. So here for our ligand we've now painted all the carbons pink, so the ligand will stand out from the rest of the protein. The ligand obviously isn't pink, but it's just a useful way to display it. So this is the sort of thing I'm trying to get across, is that our actual data is just a list of atom positions, but what we do is we display it in interesting ways, so we can ask particular questions about the protein structure that we're interested in. So that might make a nice picture of the active site. There's a couple more things I would just like to show you. This might take slightly longer. Okay, so show surface. Look here, it's progressing. Takes a bit of time, so it takes the computer energy to show a surface. And so this surface that it's generating is to imagine that if you sort of rolled a ball over where all the atoms were and tried to work out where you could find little pockets, this is what the surface would look like. So you can see here, you can see little cavities. There's another little cavity here. This is still coloured in the same way that we had it coloured before, so the carbon atoms are all coloured in this light blue, the nitrogens in the dark blue and the oxygens in the red. So you can see it's got a sort of speckled approach. Sorry, speckled, I don't know what you call it. Speckled look. The little oranges you can see are sulfur atoms. And you can actually see, it's quite interesting here, that these little stars, which are the waters, you can see how they fit in the pockets, which is why they've probably bound tightly to the protein, because they've sort of gone into a pocket and they've bound there. So we can try and zoom in. This may or may not work. Probably not, because we've probably centered in the wrong place. We've got inside. Okay, so sometimes you can, and it's quite tricky, sometimes you can get inside the protein to look at the surface there. So like the surface of the active site, which the ligand is sitting in. I have made a picture like that, but I think I'm not going to be able to just do it so simply, and I probably need to check much more carefully where the ligand was and stuff. But you can also see inside surfaces. It's just you need to plan it a bit more carefully. You can also generate electropotential of your protein, and this also takes the protein quite a lot of thinking time, so we'll see if it works or crashes or not. And so what this does is it also shows the surface, but rather than just showing you the colours of what you've previously painted the atoms, you know, light blue, red and blue, it tries to just colour the molecule by potential charge. So if you've got an arginine, it's going to be positively charged, and so what it will do is it will put a blue splodge where the arginine is because it's positively charged, or if you've got an aspartate that's negatively charged, it will put a red splodge where the aspartate is. And so you can look for sort of the charge distribution of your protein without worrying about all these carbonyl oxygens just sticking out, giving you red blobs. but I don't know if it's going to do it. Aha, here we go. So this is another surface, but as I say, coloured by charge, and this is it's an approximate calculation. It's not sort of gospel, but it's quite helpful, you know. So maybe if you think you've got a positively charged ligand that might fit in a negative pocket, then you would look for a red patch like this maybe on your protein. And for most globular proteins, you will see that they are generally white with these red and blue patches on them to suggest different charged regions, whereas if you had a membrane protein, you would expect to see a particular pattern in having less charge where the protein was interacting with the membrane. This is also a useful view to ask particular questions about your protein. And finally, if we can just get rid of this, I would just like to show you a B factor view, which is like this. So apart from our atom positions, which are the main result of our experiment, the final thing that we can look at is the flexibility of any particular atoms in our crystal, and it's really a measure of the disorder. So how certain are we that this atom is actually here? And some atoms are more disordered than others, and so we assign them this B factor. And what this view does is it colours atoms by B factor. So where atoms have low B factors, that means we're very sure of where they are, they have low disorder, then they're coloured in this blue colour. So it's sort of a rainbow scale, and green is somewhere in the middle. And where you have these red and yellow atoms in these loops around here, these atoms are much more disordered. We're less sure of exactly where they are in the crystal, and so we can represent them as sort of like a blur around here. So this atom is somewhere in this volume. So this is a B factor putty display, with the red meaning high B factor, lots of disorder, and the blue low B factor, not much disorder. So this is just a tour of some of the ways we can look at crystal structures. Gives you an idea of how we can go about analysing what our structure means, and shows you how people create all those pretty pictures that you probably see in your textbook, and how that relates to the actual information that we get out of our experiment, which is that boring text file with lots of atom positions in.